Hi, this is Heather Peterman. This is the biochemical testing for microbial identification lab. I have already cleared my work area, and I allowed the tubes of the MRVP broth to warm to room temperature. I have washed my hands with soap and water. I have my safety gloves, face mask, apron, and goggles ready. I've disinfected my workspace with 10% bleach solution. And I've labeled 2 of my vials here with MRVP S. epidermis and MRVP broth E. coli. And now I have placed 2 dropper pipettes into a disposable cup of alcohol right here. And I'm going to now light my candle and gather the active E. coli culture vial, and then I'm going to remove the lid on the vial and pass it over the flame. Okay, so I'm now taking the I took the lid off the E. coli MRVP broth tube, and now I'm going to flame the lip of the tube. And then I'm going to take my pipette. I'm going to place it into and I'm going to draw up a small amount of the broth. There. Okay, so I placed the 4 drops of the active culture into the MRVP tube, and then I put the pipette into undiluted bleach. Then I flamed the lip of the inoculated MRVP broth vial, and I screwed on the cap. And then I'm now going to just repeat those same steps with the S. epidermis culture. So it has been 48 hours since my since I put my cultures into incubation. Unfortunately, I am not seeing much of anything, so I am going to continue to incubate these for another 24 hours. Okay, so now for the second, the part 2, I let my MRVP broth incubate for 48 hours. I didn't see much bacterial growth, so I did it for another 24 hours. Now here I am. I wiped down my work area again. I have all my safety equipment on, and I have sterilized these 4 test tubes by rinsing them in alcohol and allowing them to dry. And then I labeled 2 of them E. coli and 2 of them S. epidermis. And now I'm going to pour half of the incubated MRVP broth, the ones that are labeled E. coli, into the 2 E. coli test tubes. And then I'll repeat the same for the S. epidermis MRVP broth.